Today I want to discuss the dengue vaccine dilemma, and I also want to discuss how this may lead to a Zika vaccine dilemma based upon the same kind of problems. Keep in mind that dengue has been around a lot longer than the Zika virus in terms of medical research and its effect on the human population. But dengue continues to be a very serious viral threat to large fraction of the human population. There may be as many as 390 new cases of dengue each year, and a small but serious fraction of those cases leads to what is called serious dengue fever. Now, serious dengue fever is probably related to a transient increase in vascular permeability. In other words, your plasma gets out of your vessels and perhaps other things get into your blood vessels. And this may lead to circulatory compromise, shock, and death. There's been quite an effort to develop a dengue vaccine, and it's actually been going on for quite a long time. And of course, one of the reasons why you would want to employ wide-scale dengue vaccination is that you would control the disease. But dengue vaccine development has been slow and very difficult. And the reason for this is the vaccine development is hindered by the complex immune response to the dengue virus and the difficulty in eliciting concomitant protection against its antigenically distinct serotypes. Why is this important? Because your first infection with dengue with one of the serotypes might be fairly benign, but when you have the second infection with a different serotype, then you're at risk of these major serious dengue symptoms. Now, the only licensed dengue vaccine, I believe, still is the CYD TDV. Now, you'll notice in this article that particular abbreviation is not actually defined, but I figured out that it stands for chimeric yellow fever dengue tetravalent dengue vaccine. That's what it means. Chimeric yellow fever dengue means that a previous vaccine for yellow fever was modified, and the coat proteins, that is the proteins that are facing the outside and are the ones that perhaps produce the most crucial immune responses, are replaced. The yellow fever versions of those coat proteins are replaced with the dengue version. And tetravalent means that they did this for each of 4 serotypes that were prevalent when they started making the vaccine. Of course, there may already be another serotype, and of course there's no way to know whether there may be additional serotypes. They started out with an attenuated yellow fever vaccine called 17D, and then, as I said, they replaced the corresponding genes, the E envelope protein gene and the pre-membrane protein gene from each of the 4 dengue serotypes into this chimeric yellow fever dengue vaccine. During the course of the phase 1 and phase 2 and phase 3, and especially the large phase 3 trials, which were conducted in 5 Asian countries, there was a lot of data collected, and they looked through all of the data, and in a pooled analysis of follow-up at 25 months after the first dose, vaccine efficacy against symptomatic dengue was 60% for all participants and 66% for those 9 years of age or older, and 45% for younger than 9 years of age. Now, this may seem like a bit low efficacy, and it leads to additional issues because remember, if the efficacy is suboptimal, then you may have put the recipient of the vaccine into a similar immune state as those who have received one of the serotypes and then receive a second serotype a while later after the immune response has attenuated. So right now, because of these kinds of issues, the vaccine is licensed in Paraguay, Mexico, Brazil, El Salvador, Costa Rica, and the Philippines. Oh. And it's licensed for pre-adolescents, adolescents, and adults from 9 to 60 years of age in those countries. Now, what this article is all about is that the World Health Organization convened a strategic advisory group of experts with the designation SAGE, who reviewed this evidence and recommended that the countries consider introduction of the vaccine only in settings with high endemic levels, as defined by a seroprevalence of at least 70% in the target age group. Now, what does this mean? This means that in a country like the United States, where the seroprevalence is very low, except in certain areas of the country, and including Puerto Rico and Hawaii and other areas, you shouldn't be using this vaccine. And this is the only licensed vaccine. And this means you can't contain the disease by vaccinating a big majority of an overwhelming majority of the people that are exposed to the mosquitoes and therefore to the virus. But this recommendation is certainly a split, has resulted in a split in the dengue community, and among those who support and those who challenge these conclusions. But why is that? Well, the reason is that if you are seronegative, the chances are great that you will get an increased risk of hospitalization from dengue if you receive the vaccine. So this means that really you shouldn't take the dengue vaccine unless you're seropositive. Then you would have a reduced chance of going into the hospital with a dengue infection. So this is very depressing news, as you may imagine. And anytime a vaccine does harm instead of good for any major fraction of the recipients, then one has to really worry about this. Well, now I'm going to turn on to the other possibility, which is that this kind of problem may occur with Zika. Right now, we don't know whether the very serious problems from Zika infections that are noted in Brazil, which include neurological and problems with unborn fetuses that result in smaller head size, microcephaly, are due to the immune status of the person who received the Zika infection. Could it be that one or more of the strains of dengue, which is fairly closely related to Zika, could elicit a dysfunctional immune response that results in these serious consequences? Well, we just don't know. Now I'm going to get on my high horse and pontificate. As a very well-trained scientist with a lot of experience in an institution doing preclinical cancer research, but really not much of a experience in vaccine research, when I start reading some of the preclinical papers, these are papers that are done on tissue culture samples and also on laboratory animals, I find that the investigators do not seem to list all of their assumptions. And in fact, their conclusions, in my opinion, on some of these papers, but not all, is that the conclusions are really very weak. Now, as someone from the cancer center community, preclinical cancer center community, I have to be careful because there have been reports that even preclinical cancer research is not reliable. One pharmaceutical company that attempted to rely upon the literature found when they tried to reproduce, I think it was about 10 or so different studies, they found an appalling inability to reproduce these studies. This needs to change, and it needs to change right now in the Zika research community. So I'm in favor of a fairly radical change in the way at least a portion of NIH funds research grants. I would like for those grants to be based upon two things: reproducibility of the results and significance. Now the problem is reproducibility, because it costs money to reproduce a study. But I believe that in the dengue and Zika research arena, NIH should start a small program in which a group of individuals, perhaps well-trained undergraduates even, will read papers, come to conclusions about what the assumptions are, then read additional medical and scientific literature, and come to some sort of idea of what things need to be investigated further. And NIH should fund summer research projects for those individuals who've come up with these ideas in labs which could reproduce the original results and perhaps even extend them. I think this fairly low-cost effort would really result in a very big payoff in terms of reliability of the research. Now, once you got a undergraduate summer program to reproduce your results, then NIH should know about this and provide merit awards to those whose results are of interest and are reproducible, even if it's determined by undergraduate research efforts, but not punitive, because we're not going to punish labs whose results weren't reproduced, but we should at least try to give a carrot, not necessarily a stick, in this new possible program. Okay, please tell me what kind of comments you have about this. If you're interested in supporting my own research efforts, I'm trying to start a biotech company to address these issues. Please contact me at zika.update@gmail.com and indicate if you're interested in giving financial support or moral support, and I'll try to reply back to you. And also, if you give me your telephone number, I'd be willing to talk about any particular scientific issue that comes to your mind. Okay, thank you so much for your attention.